This is a continuation of a musculoskeletal system. In this video, we will cover bone formation or ossification or osteogenesis. Osteogenesis is defined as a formation of bone. Ossification, also called osteogenesis or bone mineralization, is a process of bone formation by osteoblasts. Osteogenesis is the formation of a bone by the activity of osteoblasts and osteoclasts and the addition of minerals and salts. During the first 8 weeks of development, the skeleton is cartilaginous. The bones increase greatly in size as the fetus develops and throughout childhood. Bone growth continues through adolescence. The process of replacing other tissues with bone is called ossification, which involves the deposition of calcium salts. In embryos, ossification as well as osteogenesis leads to formation of the skeleton. Later in development, bone growth occurs until early adulthood. Throughout life, the bones can become thicker. Ossification in bone remodeling is a process of laying down new bone materials by cells named osteoblasts. It is synonymous with bone tissue formation. Calcification is often confused with ossification. Ossification is distinct from the process of calcification, whereas calcification takes place during the ossification of bones. It can also occur in other tissues. In the early stages of embryonic development, the embryo skeleton consists of fibrous membranes and hyaline cartilage. By the 6th or 7th week of embryonic life, the actual process of bone development, ossification or osteogenesis, begins. Bone is a replacement tissue, that is, it uses a model tissue on which to lay down its mineral matrix. For skeletal development, the most common template is cartilage. During fetal development, a framework is laid down that determines where bones will form. This framework is a flexible, semi-solid matrix produced by chondroblasts and consists of hyaluronic acid, chondroitin sulfate, collagen fibers, and water. As the matrix surrounds and isolates chondroblasts, they are called chondrocytes. Unlike most connective tissue, cartilage is avascular, meaning that it has no blood vessels supplying nutrients and removing metabolic waste. All of these functions are carried on by diffusion through the matrix. This is why damaged cartilage does not repair itself as rapidly as most tissues do. Throughout fetal development and into childhood growth and development, bone forms on the cartilaginous matrix. By the time a fetus is born, most of the cartilage has been replaced with bone. Some additional cartilage will be replaced throughout childhood, and some cartilage remains in the adult skeleton. Bone growth continues until approximately age 25. Bones can grow in thickness throughout life, but after age 25, ossification functions primarily in bone remodeling and repair. There are two processes or pathways resulting in the formation of a normal healthy bone tissue. They are intramembranous ossification and endochondral ossification, but bone is the same regardless of the pathway that produces it. Heterotopic ossification is a process resulting in the formation of a bone tissue that is often atypical at an extraskeletal location. Now we will see intramembranous ossification. Ossification begins approximately 6 weeks after fertilization in an embryo. Before this time, the embryonic skeleton consists entirely of fibrous membranes and hyaline cartilage. The development of bone from fibrous membrane is called intramembranous ossification. Intramembranous bonds originate between layers of connective tissues that are sheet-like in appearance. Intramembranous ossification is the direct laying down of bone into the primitive connective tissue called mesenchyme. Ossification begins as mesenchymal cells forms a template of the future bone. The process begins when mesenchymal cells in the embryonic skeleton gather together and begin to differentiate into specialized cells. Some of these cells will differentiate into capillaries, while others will become osteogenic cells and then osteoblasts. Although they will ultimately be spread out by the formation of a bone tissue, early osteoblasts appear in a cluster called an ossification center. Osteoblasts cluster together to create an ossification center. Bone-specific cells, known as osteoblasts, then start secreting the extracellular matrix osteoid, a non-mineralized collagen proteoglycan matrix or non-calcified matrix that has the ability to bind calcium. So, secretory collagen and bone matrix proteins form a complex interwoven network known as osteoid, creating the foundation of a bone. As calcium binds to the osteo, the matrix calcifies or hardens within a few days as mineral salts are deposited on it, thereby entrapping the osteoblast within. Once entrapped, the osteoblasts become osteocytes. As osteoblasts transform into osteocytes, osteogenic cells in the surrounding connective tissue differentiate into new osteoblasts. Osteoid eventually undergoes calcification and mineralization to form lamellae or layers in the bone matrix. This process constitutes the development of the hardened material recognized as mineralized bone. As osteoblasts continue to secrete osteoid, the non-mineralized portion of the bone or osteoid continues to form around blood vessels, results in a trabecular matrix, leading to the formation of a trabecular or cancellous or spongy bone. These blood vessels will eventually develop into red bone marrow. The spongy bone is remodeled into a thin layer of a combat bone on the surface of the spongy bone. Osteoid secreted around the capillaries results in a trabecular matrix, while osteoblasts on the surface of the spongy bone become the periosteum. Mesenchymal cells on the bone surface form a membrane known as the periosteum. The periosteum then creates a protective layer of combat bone superficial to the trabecular bone. The trabecular bone crowds nearby blood vessels, which eventually condense into red marrow. During intramembranous ossification, combat and spongy bone develops directly from sheets of mesenchymal or undifferentiated connective tissue. The flat bones of the face and skull, most of the cranial bones, the mandible, hip bone, and the clavicle are formed via intramembranous ossification. These bones, also called dermal bones, begin development when unspecialized connective tissues form at the sites where future bones will be developed. Intramembranous ossification begins in utero during fetal development and continues on into adolescence. At birth, the skull and clavicle are not fully ossified, nor are the sutures of the skull closed. This allows the skull and shoulders to deform during passage through the birth canal. The last bones to ossify via intramembranous ossification are the flat bones of the face, which reach their adult size at the end of the adolescent growth spurt. Now we will see endochondral ossification. The skeleton of a human embryo before 8 weeks of justification is made of fibrous membrane and hyaline cartilage. At week 8, bone tissue begins to develop, replacing most existing cartilage or fibrous structures over time. For example, the long bones are first formed of hyaline cartilage and later replaced by bony tissue that becomes combat bone. This process begins with the diaphysis and ends with the epiphysis of each long bone. This process of bone formation is called endochondral ossification. Cartilage is also referred to as endochondral bone. So, endochondral ossification is the process of bone development from hyaline cartilage. This requires a hyaline cartilage as a precursor. All of the bones of the body, except for the flat bones of the skull, mandible, and clavicle, are formed through endochondral ossification. Endochondral ossification is the formation of long bones and other bones. In endochondral ossification, bone develops by replacing hyaline cartilage. Cartilage does not become bone. Instead, cartilage serves as a template to be completely replaced by new bone. Endochondral ossification takes much longer than intramembranous ossification. Bones at the base of the skull and long bones form via endochondral ossification. In a long bone, for example, at about 6 to 8 weeks after conception, some of the mesenchymal cells differentiate into chondrocytes, that is called cartilage cells, that form the cartilaginous skeletal precursor of the bones. Soon after the perichondrium, a membrane that covers the cartilage. In the last stage of prenatal bone development, the centers of the epiphysis begin to calcify. Secondary ossification centers form in the epiphysis as blood vessels and osteoblasts enter these areas and convert hyaline cartilage into spongy bone. Until adolescence, hyaline cartilage persists at the epiphyseal plate or growth plate, which is the region between the diaphysis and epiphysis that is responsible for the lengthwise growth of long bone. There are two centers of ossification for endochondral ossification. They are the primary ossification center and the secondary ossification center. Now we will see primary ossification center. Nearly all bones below the base of the skull except the clavicles are formed by endochondral ossification. At month 2 of gestation, hyaline cartilage previously formed are used as models for actual bones. Endochondral ossification is more complicated than intramembranous ossification because hyaline cartilage is broken down while ossification is occurring. For long bones, the center of a hyaline cartilage shaft is where ossification begins. Blood vessels form in the perichondrium that covers a hyaline cartilage bone model and convert it to a vascularized periosteum. As nutrients become more plentiful, underlying mesenchymal cells specialize to become osteoblasts. This becomes the basis for ossification. Bone colloids form around the diaphyses of the hyaline cartilage models as osteoblasts secrete osteoid against the hyaline cartilage diaphyses. They enclose it in a collar-like structure known as the periosteal bone collar. In the center of the diaphysis, the cartilage calcifies, developing cavities. Chondrocytes in the shaft enlarge, which leads to the surrounding cartilage matrix to calcify. Because this matrix cannot be penetrated by diffuse nutrients, chondrocytes die. The matrix deteriorates, opening up cavities. The bone collar stabilizes the hyaline cartilage model. In other locations, the cartilage is still healthy and growing quickly, which lengthens the cartilage model. By the third month, the cavities are invaded by elements collectively known as the periosteal bud. This contains a nutrient artery and vein, red marrow elements, nerve fibers, osteoclasts, and osteogenic cells. The calcified cartilage matrix is partially eroded by the osteoclasts. The osteogenic cells become osteoblasts, secreting osteor around calcified fragment of hyaline cartilage. This forms a bone-covered cartilage trabeculae. Therefore, an early version of a spongy bone develops in the long bone. Osteoclasts break down the new spongy bone as the primary ossification center enlarges. A medullary cavity is opened in the center of the diaphysis. From week 9 until birth, the quickly growing epiphyses are made only of cartilage. The hyaline cartilage models continue lengthening via the division of viable cartilage cells at the epiphysis. Down the length of the shaft, cartilage calcifies, erodes, and is replaced by spiker bone stretches on epiphysis surfaces that face the medullary cavity. At birth, most long bones have a bony diaphysis that surrounds spongy bone remnants, a medullary cavity that is widening, and two epiphyses made of cartilage. In long bones, bone tissue first appears in the diaphysis. Chondrocytes form a template of the hyaline cartilage diaphysis. Responding to complex development signals, the matrix begins to calcify. As more matrix is produced, the chondrocytes in the center of the cartilaginous model grow in size. As the matrix calcifies, nutrients can no longer reach the chondrocytes. This results in their death and the disintegration of the surrounding cartilage. So, the calcification prevents diffusion of nutrients into the matrix, resulting in chondrocytes dying and the opening up of cavities in the diaphysis cartilage. Blood vessels invade the cavities, not only enlarge the cavities but also carrying osteogenic cells with them, many of which will become osteoblasts. Osteoblasts and osteoclasts modify the calcified cartilage matrix into spongy bone. Osteoclasts then break down some of the spongy bone to create a marrow or medullary cavity in the center of the diaphysis. As the cartilage grows, capillaries penetrate it. This penetration initiates the transformation of the perichondrium into the bone-producing periosteum. Here the osteoblasts form a periosteal collar of a combined bone around the cartilage of the diaphysis. A perichondrium layer surrounding the cartilage forms a periosteum around the bones, which generates osteogenic cells that then go on to make a collar that encircles the outside of the bone and remodels the medullary cavity on the inside. The periosteum assists in attaching the bone to surrounding tissues, tendons, and ligaments. The bone continues to grow and elongate as the cartilage cells at the epiphysis divide. Chondrocytes multiply and form trabeculae. Cartilage is progressively eroded and replaced by hardened bone, extending towards the epiphysis. By the second or third month of fetal life, bone cell development and ossification ramps up and creates a primary ossification center, a region deep in the periosteum called where ossification begins. The nutrient artery enters via the nutrient foramen from a small opening in the diaphysis. It invades the primary ossification center, bringing osteogenic cells, osteoblasts on the outside, osteoclasts on the inside. The canal of the nutrient foramen is directed away from more active end of bone when one end grows more than the other. When bone grows at the same rate at both ends, the nutrient artery is perpendicular to the bone. Most other bones, for example vertebrae, also have primary ossification centers and bone is laid down in a similar manner. Now we will cover secondary ossification center. Secondary ossification centers develop in one or both epiphyses just before or just after birth. Usually, secondary centers form in both epiphyses of larger long bones and in smaller long bones. Usually, only one secondary ossification center forms. The central cartilage of the epiphysis calcifies and deteriorates. Cavities open, allowing a periosteal bud to enter. Bone trabeculae appear similar to how they appeared in the primary ossification center. Short bones develop differently in that only the primary ossification center is formed, and most irregular bones have several distinct ossification centers from which they develop. Secondary ossification is very similar to primary ossification except for interior spongy bone being retained and the lack of a medullary cavity being formed in the epiphysis. Hyaline cartilage remains only on the epiphysis surface as articular cartilages and at the junction of diaphysis and epiphysis, forming epiphyseal plates when secondary ossification is complete. Flat bones are not formed in the same way as long bones. Flat bones develop from fibrous connective tissue membranes formed by mesenchymal cells that are replaced by spongy bone and then combined bone in a process is called intramembranous ossification. The produced bones are also referred to as membrane bones. In the embryonic skeleton, membranes and cartilages allow for mitosis. Examples of flat bones formed via intramembranous ossification include the frontal, parietal, occipital, and temporal bones of the skull and the clavicles. Ossification begins at about 8 weeks of gestation. When the bones are growing, the diaphyses meet the epiphysis at a structure called the epiphyseal plate. It is made of 4 cartilage layers: reserve cartilage, proliferating hyperplastic cartilage, hypertrophic cartilage, and the calcified matrix. Endochondral bones begin as cartilaginous masses that are eventually replaced by bone tissue. These bones develop from hyaline cartilage that is shaped similar to the bones they will become. They grow rapidly at first and then begin to change in appearance. When spongy bones begin to replace the original cartilage, a primary ossification center is created with bone tissue developing outward toward the ends of the structure. While these deep changes are occurring, chondrocytes and cartilage continue to grow at the ends of the bone, which increases the bone's length. At the same time, bone is replacing cartilage in the diaphysis. By the time the fetus skeleton is fully formed, cartilage only remains at the joint surface as articular cartilage and between the diaphysis and epiphysis as the epiphyseal plate, the latter of which is responsible for the longitudinal growth of bones. After birth, this same sequence of events such as matrix mineralization, death of chondrocytes, invasion of blood vessels from the periosteum, and seeding with osteogenic cells that become osteoblasts occurs in the epiphyseal regions. And each of these centers of activity is referred to as secondary ossification center. Eventually, secondary ossification center appear in the epiphysis, forming more spongy bone. Secondary ossification mostly occurs after birth, except for distal femur and proximal tibia, which occurs during 9th month of fetal development. The epiphyseal arteries and osteogenic cells invade the epiphysis, depositing osteoclasts and osteoblasts, which erode the cartilage and build bone respectively. This occurs at both ends of long bones, but only one end of digits and ribs.